Assalamualaikum. Halo mahasiswa, selamat datang di video pembelajaran kali ini, di mana kita akan menjelajahi topik menarik seputar kloning hewan. Kloning atau klonasi merupakan teknik penggandaan gen yang menghasilkan turunan yang sama, baik dari segi hereditas maupun penampakan dengan induknya, pada organisme baik tumbuhan, hewan, maupun manusia. Atau secara singkatnya, kloning adalah proses pembuatan organisme baru yang memiliki materi genetik dengan induknya. Namun, dalam kehidupan sehari-hari, kloning masih menimbulkan pro dan kontra. 70 tahun yang lalu, kloning adalah karya yang digunakan terutama dalam penelitian dan aplikasi celana. Kini, kloning bisa dilakukan pada semua jenis makhluk hidup, terutama manusia. Domba Dolly adalah hewan tiruan paling terkenal di dunia dikloning melalui teknik transfer inti sel somatik atau SCNT. Tapi itu bukan yang pertama. Banyak hewan termasuk katak, tikus, domba, dan sapi telah dikloning sebelum Dolly. Ada beberapa teknologi yang digunakan dalam proses kloning hewan. Yang pertama yaitu Somatic Cell Nuclear Transfer atau SCNT. Yang kedua yaitu Pluripotent Stem Cell Nuclear Transfer. Yang ketiga yaitu cloning embrio. Langkah-langkah yang dilakukan kloning hewan. Yang pertama, persiapan sel induk. Selanjutnya, pengosongan sel telur. Selanjutnya, penggabungan sel induk dan sel telur. Selanjutnya, stimulus pembelahan. Kemudian, implantasi embrio. Selanjutnya, keberhasilan kehamilan dan kelahiran. Dan perawatan dan perkembangan.
This animation outlines the process the Roslin Institute scientists followed to create Dolly, the world's first mammal to be cloned from cells taken from an adult Dorset ewe. To help you follow the process, we have labelled the parent sheep, sheep 1 and sheep 2. Note that sheep 1, the ewe to be cloned, has a white face, a fact that was used to help check that cloning was successful. The first step involved taking cells from the udder of sheep 1 and culturing them in a gel to create a batch of identical cells. In the course of culturing these cells, they were deliberately deprogrammed to let these cells regain their potential to become totipotent, to form any cell needed in the future clone, and not simply another cell from a ewe's udder. This single achievement marked a significant breakthrough. Meanwhile, an egg or ovum was removed from sheep 2, a black-faced female Scottish ewe, to be prepared for implantation. Using techniques developed in human fertilization treatments conducted at a microscopic level, the nucleus from the sheep 2's ovum was removed, or enucleated, to make sure that none of sheep 2's DNA and genotype could be passed on to the next generation. Once the ovum had been enucleated, it was then fused with a cell from the sheep 1 culture of deprogrammed udder cells. In effect, emulating the process of a sperm entering the egg, but with both copies of the chromosomes coming from the same parent sheep, sheep 1. So the newly fertilized egg is genetically identical with this single parent. Once fusion had taken place, chemical factors in the egg are released to stimulate cell division. Once the cells had multiplied sufficiently to form the makings of an embryo, the new cell cluster was transplanted into the womb of a third sheep, which we will call sheep 3, for a 21-week gestation period. In total, 277 attempts were made to fuse the adult cells with enucleated eggs. Only one survived. Dolly was born on the 5th of July 1996 by caesarean section and was found to be genetically identical to sheep 1, from whose udder her DNA originated. Dolly lived until the 14th of February 2003, when she was put down suffering from a progressive lung disease.
Terima kasih telah menyempatkan waktu untuk menonton video pembelajaran ini. Semoga menambah wawasan baru dan pengetahuan yang berguna. Ingatlah untuk eksplorasi lebih jauh dan mencari tahu lebih banyak tentang kloning hewan ini. Semoga video pembelajaran ini membuka pintu pengetahuan lebih lanjut.